obviously before we went on our break, we were working on our Gel Electrophoresis Lab, and we put our DNA samples into the thermocycler. We were looking to get those PCR products. If you remember, the job of the the job of the thermocycler is to make more of the DNA sample so that we can run the gels. So I have set up the gels, sort of, and I am going to try, we're going to see how it goes, but I'm going to try to show you how to load the gels and see how it works. So I have my pipette tips. I've got my pipette. I have my gel. The gel here is still in the casing right now, but I'll scooch that out in a minute. This is the like the Jello mold that we use to pour the gels, and I haven't taken the comb out yet so that I have the wells where I'll put the DNA samples in. What I would like to try to do, and we'll see how well you can see it, is I want you to be able to see the overhead view of me loading, and I kind of, if you can, I'm going to try to turn the camera and see if I can show you the side view as well. We'll see how it goes. But I'm going to be using loading dye because I don't have the DNA samples. If you remember from my last video, I said I'm trying to salvage them. We'll see if they're still good when we come back to school. We'll try it, but I'm not sure if it'll work. But what I did bring home was containers of loading dye. So, of course, I also dropped them so you can see all of the droplets in the top. And I didn't bring a centrifuge home, so I can't spin them. But whatever, it's fine. I'm going to just try loading the gels just with loading dye, just so that you can see the loading dye should be denser. So it should sink down into the wells so you can kind of see how it sits down in the wells. And then obviously I'm not going to hook it up to an electrical source because there's no DNA in it. I'm not actually going to run it. But we're going to give it a shot. Okay? So the first thing I'm going to do is zoom in just a smidge here. First thing I'm going to do is take the comb out of the gel, and then I'm going to take the top off this. This actually should go the other way. If you can see on the side of the chamber, you can see that yellowish arrow that points this way, which means we want the wells on this side because the DNA is going to run this way. So I like to have the wells on the top when I'm loading, so I'm going to put it that way. So then this kind of gets gently, hopefully, gently lifted out of the cast. And obviously you can't feel it, but maybe you can see the wells. I get them nice and close. You can see them. And across my finger, they are like rough, so you can see that they're raised along the edge here. They're actually teeny tiny little holes, but they do not go all the way through. So it's smooth on the back, and the holes don't go all the way through. So we put the gel in. Typically we would cover the gel with what's called a buffer. To avoid bringing all sorts of chemicals home, I did not bring the buffer home. So we are going to give this a whirl with water. Let's see what happens. I might leave the light off because I have a good amount of overhead light. Let's see how it works. So before I load, I'm going to, normally it would be buffer, but we're going to do water. Let's see if I spill it all over my table. I would cover the gel with buffer. Buffer is basically a liquid that has electrolytes and other compounds in it that will help it conduct electricity better. And since normally we would be hooking this up to an electrical source, that is exactly why we want to have it conduct electricity better. So now I'm going to turn, you don't need to see my face anymore, I'm going to turn this camera so maybe, hopefully, you can see. How's the view? Oh, that's not bad. Now the next trick will be to keep my arm out of the way while I'm doing this. But I'm going to take the pipette tip. I'm going to set it for, I think it was 20 microliters. I'm going to set it for 20 microliters. I put my pipette tip on. I have my container of loading dye. The loading dye for us is orange. If you remember from the video on Wednesday that you watched the Ed Puzzle video, they talked about, I think her example was like a pink, a yellow, and a green, and I made a note there that ours would have been orange. They come in different various colors. It is super annoying that there's so much so many droplets in the top there, but I didn't bring home a centrifuge, so I can't spin them. So hopefully there's enough in here. Put that there. Scooch myself over. So for me, it's important that I set my elbows on the table. The other trick here will be to set my elbows and not block your view. Okay? But it's pretty important for me to set my elbows. So I'm going to get my 20 microliters. Here it is. You set your elbow. See if I can scoot it even closer here. Okay. So I'm going to have my right elbow is down. You can see it right here on the far side of the gel. My left elbow would normally go right here, which obviously is not helpful for you to see the video. So I'm going to try to do it a little bit differently. But basically what you're going to do is you take the pipette tip, you go down in, and it's okay that it leaks a little bit, down into one of the wells. You release the fluid all the way through that second stopping point, and you hold until your pipette tip is out of the liquid, and then you release. If you release while you're still under the buffer, you're going to suck some of it back up. So that actually wasn't too bad. I always tell students when I'm demonstrating this how absolutely terrible I am at this. Some people are really good at it. Some people are really not good at it. I like to think that if I regularly worked in a lab, I would be much better at it. But since I don't obviously do it very often, I don't do a very good job. So you could see that I had a little bit of overflow when I first went into the liquid. So let me do it one more time to make sure that you can see. Gently put the tip down into the well. If you go too far down, you will poke a hole right in the well. My thumb is still down. You can see that my thumb is still down, and now that the pipette tip is out of the liquid, now I will release. Okay? So this is what we would have done, and we would have filled it up. So we would have had an allele ladder here. Then we would have had our crime scene DNA, our suspect 1, suspect 2, suspect 3, and suspect 4, and we would have had a couple of extra wells because sometimes we poke holes in them accidentally, and then this way we have an extra one, we just scooch it over one. So that's basically the technique on how you would load these gels. After you load the gels, then you can come back to looking at my face if you'd like. After you load the gels, then you would take your top, and obviously you gotta line up the colors, the black and the red. Go with the, hold on, let me zoom out here. There we go, the black and the red. Go with the black and the red. You put it on. In class, I would have had you put a piece of tape on the top to tell you, so we knew whose was whose. And then we would take the plugs and plug them right into the power pack there that we looked at before we went on break. So the goal here will be to hopefully get a chance to look at the rest of the lab and see if we can maybe answer some more of those questions even without actually having done the lab. Okay? Alright, so if you wouldn't mind taking out your lab, it's the yellow lab that we started in, would have been Monday we even started it, I think, so we did some of the pre-lab stuff last Monday, the 9th, and then we did the PCR part on Wednesday, the 11th. If you're looking through your lab, it's a yellow lab packet. Mine obviously is white because I just have the original at home. You already should have done questions on page 34 and the questions on page 37. So we're going to start on page 40, which looks like this. Excuse me. Okay, page 40 has an example of a gel at the bottom, and it says lesson 2, electrophoresis of the PCR products. I feel like it's blurry. Does anybody else feel like it's blurry? Hold on. And I like how I ask as though you guys are going to answer while I'm filming in my house. You have it in front of you, so hopefully it'll be. Whoa, that's much. How about this? That's not bad. Okay, so you've completed your PCR amplification. However, at this point, you can't actually tell whether or not you have PCR products. To do this, you must sort your PCR products using the gel electrophoresis and then visualize them using a DNA stain. Since the DNA is negatively charged, it can be separated using an electrocurrent. In fact, electrophoresis means carry with current. In Agarose Gel Electrophoresis, DNA is placed in solidified agarose, which forms sieves containing pores that vary in size depending on the concentration of the agarose. The higher the concentration of agarose, the smaller the pore size, and the longer it takes for the large molecules to move. This is particularly useful when you want to compare DNA molecules of different sizes contained in the same sample. Movement through the gel occurs when an electric current is applied across the gel. Since the gel is immersed in buffer, the current will travel through the buffer and the gel, carrying the negatively charged DNA with it towards the positive anode. In addition to your PCR products, you will also be running a DNA allele ladder. In the ed puzzle that you guys did on Wednesday, it was referred to as a DNA ladder, but we call it, in this lab it happens to be called an allele ladder. It's the same thing. It's just basically a standard. So it says the allele ladder that represents all of the possible alleles for the BXP007 locus. This is a reference or a marker that you can compare your PCR reactions to so you can judge their relative sizes and their identities. In the following drawing of a gel, the samples or bands seen in the first track or lane all come from the allele ladder. So if you look at the picture there, they all come from the allele ladder. These are the standard sizes of all the alleles known to occur at this locus. So there are 8 possible alleles. 1, 2, 3, 4, 5, 6, 7, 8. With the largest at the top, it's probably about 15 bases long, which is why it's near the 15, maybe even 16. Hundreds, by the way. It's in hundreds, sorry. The sizes are from the top to bottom: 1,500, 1,000, 700, 500, 400, 300, 200, and 100 base pairs. Allele names are indicated in the figure. In the next several lanes, we can see PCR products that come from DNA samples that have been tested for what alleles they carry at this particular locus. Locus, remember, is just a location on a gene. So we're looking at these sections of DNA on one particular location on a gene. As shown in Figure 12, the sample in the lane next to the allele ladder, the crime scene sample, has a genotype that corresponds with alleles 5 and 2, which makes sense because that's where the bands are. We could say that the genotype for this sample is 5-2. That might be how you write it, 5-2. Okay? For the next example, the genotype would be 7-4, and so on, 5-2, 7-2, 10-3. Okay? So that's how you read these charts here. So turning over to page 41, you have some questions to do. So it says, why does DNA, rearrange my papers here, why does DNA run or move through an agarose gel? So go back over here when you're trying to answer that question and talk about why it moves. So DNA is placed in the solidified agarose, which forms the pores. Go back to that section right there. It talks about why it is able to move. Hint, hint, it's smaller than the pores in some spots, but you need more than that, so don't just write that. Number 2, what are the 2 techniques used to create a DNA profile in this experiment? What function does each perform? So we used 2 different techniques. The first lesson that we did on Wednesday, which you guys have done, was one technique, 3-letter abbreviation. The next technique that you used after the 3-letter one, you didn't really use, but we're pretending you used, and we're hoping that when you go back to school you're going to use it, is the one that has to do with this particular lesson. Okay, 2 words, lots of letters. And what function does each perform? So tell me what was the purpose of what we did that Wednesday, and tell me what's the purpose of this thing that we aren't really doing but want to do and hopefully will do, and you're seeing me kind of do. Okay? Number 3, what is an allele ladder? What is its function in the DNA profile? That talks about that right here. Yep, the DNA allele ladder that represents all the possibles. So there's your information for that. And then what is required to visualize the DNA following the gel electrophoresis? That is not on this page, but the only way you will be able to show or see your bands is if you stain it afterwards. So you will see that what we call the dye front. So as the dye moves through the gel, you will see a dye front, but you won't actually be able to see the bands until you stain it. So the dye front is orange because that's the color of the loading dye that we use. The stain is blue. So if I can show you, I did that video portion earlier today where I loaded the gel. So now you can see, I'm going to turn the light off. Now you can see how the loading dye has sort of like spread around, and that's not even having it connected to electricity. It's just simply because it's been sitting in this water for, I don't know, too many hours. So that is why it's sort of floating in those wells there. You can kind of see the wells a little bit here. Actually, I bet you if I took the top off, it's better. There we go. Okay, so you can see the wells, and you can see, oh, that's a much better spot to lay it in. You can see the wells, and you can see that the dye has kind of floated around in the water a little bit and has stained part of the top of the gel. And again, that's just because it's been sitting here and I never attached it to an electrical source. If I had attached it to an electrical source, once I loaded it with the DNA and the gel, then you would see this orange line move this way. You wouldn't see individual bands, but you would be able to see what we call the dye front. The dye front would move this way towards the bottom of the gel. Once we were all done with it, then we would take it and stain it, and that's how we are able to visualize the actual bands. So that's what you're looking for question number 4 there. Okay, so take some time to try to figure out the answers to those questions. I kind of walked you through them already. Scooch that. Oh, don't spill, don't spill, don't spill. Okay, it'd be a terrible way to end this afternoon. Really bright. There we go, it's a little better. Then if you keep flipping through, page 42 is basically the procedure that you would have used. You want to read through that. And then it does give you a procedure for staining the gels as well. So after you run the gels, it gives you on page 33 a procedure for staining the gels, options for staining. 2 more options of staining. And then they call the last lesson that goes with this drying and the analysis of the results. We are hoping that we'll get to do that part. We may not. We'll have to see what happens. But we will worry about that and those last few questions later. So the only thing that I want you to do today for your activity is to answer the question on page 41. Okay, so I've read through it with you. I kind of know what to do. Gave you some advice on where you might find the answers to those questions. And I will just have you write them in your lab for now, with the hope that we're ultimately going to collect them. If you would like to show me, oops, I just dropped the top. If you would like to show me that you did the questions, take a picture of it and post it underneath right here in the comments, and we'll be good to go. Okay? Enjoy your Friday. Happy Friday, and have a fabulous weekend. Talk to you soon.